Okay, good morning everyone. First, we will talk about mitosis. So, what is mitosis? Well, according to this, mitosis is a process where a single cell divides into two identical daughter cells. To clarify things, mitosis is responsible for regeneration, growth, and maintenance, while meiosis is responsible for sexual reproduction. Mitosis is divided into four phases, namely prophase, metaphase, anaphase, and telophase. And now we will discuss about interphase. Interphase is not part of the stages because it is only responsible for preparation. Let's take a look at this microscopic photograph of the cell during interphase. They say that 90% of the time process are consumed by interphase. So first, let us identify the different parts of cell. So this is called the centrioles. And this one is the nuclear membrane, or also called as nuclear envelope. And this one is the nucleolus, which is centermost part of a cell. And this one is chromatids. During interphase, the nuclear membrane and the nucleolus are distinct to each other. Means they have distance towards each other, and the chromosomes are in the form of thread-like called chromatids. It is called chromatids, not chromosome, because it is not condensed yet. And as I said before, interphase only a preparation, so the parts only prepare themselves for the next true stage of mitosis, which is the prophase. Okay, during the prophase is the first true step of the mitotic process. It means that the mitosis process is first occurred in prophase. So, in prophase, these are the centrioles. Centrioles is also called as centrioles. And they are moving through different poles or areas. And this one is the nuclear membrane. As we could see in the nuclear membrane, they have distance with each other, or it has spaces, because nuclear membrane or the nuclear envelope starts to break. And as done before, it is called as the chromatids. It is now called the chromosomes. Why chromosomes? Because it has already condensed. The chromatids are now begin to form as chromosomes, and the spindles form. These are the spindles. Spindles or spindle fibers. In prophase, the chromatin condenses and turned into chromosomes, as I said before. The nuclear envelope breaks down. So you can see there are spaces. May I emphasize you the spaces between them here. It has spaces because it is begun to break down. And the spindles form at opposite poles of the cells. Spindles. Next is the metaphase. Metaphase is the second stage of mitosis, and this process, it separates duplicated genetic material carried through the cells. Okay, these chromosomes are carried duplicated genetic material inside us. It has DNA inside the chromosomes. During metaphase, the chromosomes align themselves in the middle of the cell called metaphase plate. This is called a metaphase plate. Chromosomes are aligned at the center of the poles, which is the metaphase plate. And the spindle fibers are connected through the chromosomes. Next is anaphase. Here is anaphase. In anaphase, the paired chromosomes separate and begin moving to opposite poles of the cell, just like this cell is pulled apart toward this, and this cell is pulled apart toward this. Just like when you saw a tug of war. Have you seen tug of war yet? So if you have already seen tug of war, you will relate to this of what I have said. It is very similar to this, honestly. Through this, it will cause a breakage between chromosomes, so they are pulled apart on opposite sides. Mainly, it will result in a breakage of chromosomes that were located in each pole. So these chromosomes will locate on this side, and these chromosomes will be located on this side. At the end of this stage, each pole has a complete compilation of chromosomes. Next stage is cytokinesis. Before telophase, I would like to present an additional stage which is called cytokinesis. On this stage, the nuclear membrane reforms. As we have seen, the nuclear membrane on the prophase have breakage, but today the nuclear membrane reformed. Comeback is real when it comes to nuclear membrane. And also the nucleolus, which is already lost in some stages. So this is nucleolus. It is a reform. So when it comes to cytokinesis, there is a cleavage furrow formed when there is a breakage. It's called cleavage furrow, just like, well, it speaks to itself, a cleavage. There is separation or a breakage toward them, because in anaphase it is pulled apart on opposite sides, and it results to a breakage and a forming of different cells. When it comes to cytokinesis, it has equal amount of each cell. It has nuclear membrane. Check. It has different nuclear membrane. It has nucleolus. Check. And it has also different amounts of chromosomes. As I could say, it has equal copy or replica of each parts. And next one is the telophase. The end part is the telophase, the last stage. It is the end of the stage. After cytokinesis, there is no cleavage furrow now. The cleavage furrow is gone, as you can see. In cytokinesis, if in cytokinesis has a cleavage at telophase, the cleavage is gone. It is completely separated. Yes, completely separated. It has different sides now. It has in these sides, and it has on this side. It has the same chromosomes, nucleolus, and nuclear membrane. Well, apparently, as you can see, it has the same. Yes, it has same nucleolus. Check. Nucleolus. Check. And the nuclear membrane, or maybe the nuclear membrane is missing, but it is not. It has. Check. And chromosomes, apparently is the most important part, the chromosomes. Check. So that's the end of mitosis. That's all about mitosis, and next you will be informed about meiosis. Hope you enjoyed my video. Thank you very much.